Greetings, students. The goal for today is to get a little bit cellular and biochemical here. Today's a cool day. We are going to figure out how exactly muscles do their thing. So we said very simply a day or two ago that muscles shorten. They shorten in the direction that the muscle fibers point, and as they shorten, they pull on the bones, and that causes movement. But that kind of leads to this big question: well, how? What's like the molecular mechanism for how they shorten? And that's what we're going to get at today. Before we get going, I did want to point out how do I get out of this? You may want to have your study guide handy. Again, the study guide can be found on Schoology right here. It's a PDF, and if you're able to print, great. It's not essential, but after these coloring pages that hopefully you took a look at yesterday, there's some diagrams here that will appear in my PowerPoint, and you may want to write on those, reference those. Doesn't really matter that much, but if you kind of like to have that visual, those are there for you. So how is it that muscles work? In order to get at this question, we need to zoom in on one of those spaghetti noodles. So remember we said that muscle fibers are cells, and these cells are much like a spaghetti noodle, and a muscle itself is a bundle of bundles of muscle fibers. A bundle of bundles of muscle fibers. So this is our single spaghetti noodle here, and if we zoom in on it, it looks like this, and we see that there's these smaller cylinders called myofibrils, and we're going to get to know these myofibrils a little bit better to figure out what is actually going on inside a muscle cell and how does it work. So structures, it's a cell, and so just like any cell, it has a cell membrane. That cell membrane is called the sarcolemma, and you may say, why not just call it a cell membrane? And there's kind of a rule in biology: why have one name when you can have 27? So sarcolemma it is. It's the specific cell membrane found in a muscle cell. There's also the sarcoplasm, which is a fancy word for the cytoplasm in a muscle cell. And the cytoplasm is just all the interior area, kind of liquidy goo within a cell. There's two things I want to point out that's in the cytoplasm or sarcoplasm of every muscle cell. The first is glycogen. Glycogen, you may remember from biology, is a storage form of sugar. And the significance of that is it's fuel. This is what powers your muscles. So your muscles actually store fuel right inside them as glycogen. And the second thing is this pigment. It's a red pigment called myoglobin, and myoglobin stores oxygen. And maybe you remember from chemistry that in order to burn fuel, you need oxygen. Fuel in your gas tank of a car requires oxygen to work, and so the fuel, the carbohydrates in your body require oxygen as well. So basically, muscles are storing the fuel and the oxygen to burn that fuel right inside them, which is kind of cool. There's another structure called the Sarcoplasmic Reticulum. That's this bluish stuff here, and that's a fancy word for the endoplasmic reticulum, which may ring a bell from biology last year. All you really need to know about this structure is that it stores calcium ions, and calcium is going to kind of pop up here and there, and so calcium's significant. So just kind of plant that seed. Calcium is in the mix, and it's going to be doing some stuff. So that's in that sarcoplasmic reticulum. And then the last structure I wanted to point out, you'll notice this says one sarcomere, and so that's what we're really going to zoom in on. So the sarcomere is basically the functional unit of muscle contraction. In other words, if we can understand how one sarcomere, this little structure right here, does its job, we can understand how muscle as a whole is able to do its job. And so if we zoom in on that sarcomere, here's just another picture of what it looks like, and you see it's made of these kind of alternating filaments, these kind of lightish brown to darker brown, light brown, dark brown. So we need to zoom in on that and figure out what's going on with the sarcomere, what's happening there. Here it is as it appears in a microscope. You can see these lines, and it's kind of a cool image there. So if we zoom in on the sarcomere, what we find is that there are these what are called thick filaments. They're kind of the darker black in the microscope, and the thin filaments, which are the lighter in the microscope. That's where the names come from. So the thick filaments are made of a protein called myosin. And so all this dark brown is myosin. And note that there's this little head sticking out of myosin. That's going to be important. And you can kind of see those little bumps here as we go. Those are all those little myosin heads. Those are the thick filaments. The thin filaments, on the other hand, are actually made up of 3 different proteins. The first protein is actin. Actin is these tannish colored circles right here. And you'll note there's this black circle right here. It's sort of covered up. That is a binding site for myosin. And remember, myosin is this little head. So actin has this binding site for myosin. That's the importance of that protein. And then there's this like rope-like thing. It's called tropomyosin. It's a rope-like protein that blocks the myosin binding site on actin. And I always think tropomyosin like traps myosin, tropo, trap. It traps myosin. It stops myosin from attaching and doing its thing. And then finally, troponin. And troponin is attached to tropomyosin and has a binding site for calcium. We mentioned calcium earlier. So troponin, I always think, is on tropomyosin. Tropon is on tropomyosin. So thin filaments have these 3 proteins. And then finally the Z lines. The Z lines mark the boundaries of the sarcomeres. And you can see in the microscope image why they were originally called lines, because they look like lines. In reality, they're actually like circular in structure. They're sort of the end of these, they're the shape of this end right here. So to understand how this muscle works, it helps if you look at my picture in the screen. There you go. To kind of visualize the sarcomere as your interlocking fingers. So thick filament, thin filament, thick filament, thin filament, and so on. Thick, thin, thick, thin. So alternating filaments. And then my thumbs, based on that, what would my thumbs represent? Did you say Z lines? I hope you did. It's Z lines. And so this would be a sarcomere. And so what happens when your muscle contracts is those thick and thin filaments slide past one another. And you can see that in this diagram. Here they are fairly spaced apart, and then they start to slide. The degree of overlap increases, and so boom, they are now sliding over one another. And you can imagine if they're sliding past one another, that sarcomere is going to get shorter. And what you have to remember is this is one sarcomere among many along the length of this single myofibril. This myofibril is one of many within the muscle cell. This muscle cell is one of many within the fascicle, or that bundle, and this fascicle or bundle is one of many within the muscle itself. So basically, all of the sarcomeres in all of this muscle, if they're all doing this, that is a beep ton of shortening that's happening, and the collective work of all of those sarcomere shortening is enough to like and actually bend that muscle and shorten it and pull on bones. And so the question then is, well, how does that shortening happen? So here's where, if you have your study guide, I think it's like the first page after the coloring pages, you should see this image in there. And you could write these steps down on that page or just keep putting them in your notes. But you'll notice in this picture, here's how we start. Tropomyosin is blocking those myosin head binding sites on actin. And so the first step in muscle contraction is calcium binds to troponin. When that happens, it causes the proteins to change shape, and that shifts tropomyosin out of the way and exposes these myosin binding sites on actin. So the first step is calcium comes in and basically moves this whole thing out of the way so that myosin can come in and do its job. Step 2 down here is once those myosin binding sites are revealed, the myosin head, that little head, can kind of and like attach itself to actin. And that is what is called a cross bridge. And remember, this is one myosin head among many, 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 many within even a single sarcomere. So crazy numbers if you think of a whole muscle. Step 3 then is that myosin head bends. You can see it's kind of vertical here, but here it's bent backwards. So it kind of, oops, I'm trying to do this like opposite here. So it bends. And when it bends, that's what's actually sliding the actin past the myosin. So that's what's contributing to this that's happening as these slide together. Step 4 is that ATP binds to the myosin and allows it to release the actin head. So ATP is what actually allows it to release the actin head. And then finally, the breakdown of ATP into ADP and phosphate, maybe you learned about that in biology last year. The breakdown of ATP prepares the head to do this all over again. And so basically, as the two filaments are sliding, is because of this collective action of all these heads going and just kind of binding, sliding, and letting go. Bind, slide, let go. Bind, slide, let go. Whoops, bind, slide, let go. Got that wrong. And so that's what's actually happening. Okay, and so again, that's happening just within one sarcomere, but then all along every sarcomere within this whole thing. This is a lot of these things going. And then that, of course, causes the muscle as a whole to shorten and pull on your bones. So a couple of things about this. Note that ATP is needed in order for the myosin head to release actin, right? So the binding of ATP is what lets that head release. So what happens to our muscles when we die and can no longer generate that ATP? And perhaps you've heard of something called rigor mortis. And rigor mortis is basically what happens is your muscles stiffen up and your body just kind of like just gets rigid after you die. And that's because you can't make ATP anymore. And so then these cross bridges form and they don't let go and your muscles kind of seize up in place. And that happens several hours after death. So I think I'm going to pause there and then I'll make another video for this second part. But you may have remembered the whole thing started with the release of calcium. Where did that calcium come from? Well, that's what we're going to do in the next screencast. So stay tuned.